Hello students, this is Geethanjali and today's video will be continuing with the helical model. So DNA was actually first identified by Frederick Mescher in 1869 and he named it as nuclein. Okay, but it was only until 1953 that its actual structure was deduced. So two men, James Watson and Francis Crick, okay, they used the work of Maurice Wilkins and Rosalind Franklin. They used their X-ray diffraction data and they proposed this famous double helix model of the DNA, which also included some rules given by Erwin Chargaff. So let us see what actually the double helix model proposes. So the first point, as I already said in the previous video, that the backbone of the DNA is the sugar and the phosphate. So you can see these two components are fixed, whereas the nitrogen bases differ from one nucleotide to the other nucleotide. So they basically form the backbone. The second point says that they have anti-parallel polarity. What does this mean? So they run basically parallel to each other but in opposite directions. So you can see this DNA runs from the 5' to 3' towards the right, and this DNA runs from the 5' to 3' towards the left. So this is basically the anti-parallel polarity we're talking about. So the next point mentions that the nitrogen bases of the two anti-parallel DNA, they form bonds. So you can see here, all of them have formed bonds. So the adenine always pairs with thymine, okay? And guanine always pairs with cytosine. This is a rule, okay? And adenine forms two hydrogen bonds with thymine, and guanine and cytosine, they form three hydrogen bonds with each other. And because of this, always the amount of purine is equal to the number of pyrimidines. So A plus G is equal to T plus C. And A is always equal to T and G is always equal to C. So these were basically the rules that Erwin Chargaff stated. Coming to the next point, the two chains are always right-handed in fashion. So what does right-handed in fashion means? Suppose you are holding your hand in the form of a thumbs up, okay? So you use your right hand. So in that case, the axis of the helix, okay, this axis of the helix pointing towards the direction of the thumb will give you the direction of coiling of DNA as the other fingers point. So if this is your thumb like this, okay, this is the direction of the axis of the DNA and your thumbs are folded like this. So this will be the direction of coiling of the DNA. So that is basically what right-handed fashion means. Now the pitch of a typical of our DNA, so it is 3.4 nanometers. Okay, so what exactly is pitch? Pitch is a complete turn, okay, a 360-degree complete turn. So basically from this point till this point. In this complete turn, it contains roughly 10 base pairs, okay, and that is why each base pair is distant towards the other by a fraction of 0.34 nanometers, okay, because 3.4 divided by 10, right, that's why. And the last point says that all these base pair stack over each other in one plane, okay? Whereas this particular part, that is the backbone, it actually twists around these. So that is why you can see this helical sort of shape over here. So there is one stack like this, constant stack, and basically the DNA over here, it twists around it. And this leads actually to this stability of the helical structure. Soon, Francis Crick, he proposed the central dogma of molecular biology. So, what is the central dogma? He basically deduced the direction of transfer of information. So we know that we have genes, right? Genes are nothing but our DNA sequences. So this DNA sequences help in the formation of RNA. And this RNA in turn translates itself to proteins. So the flow of information is from DNA to mRNA, that is RNA, and RNA to protein. The process of formation of RNA from DNA is called transcription. Okay, and the process of formation of protein from mRNA is called translation. And the DNA, it helps in replication by itself. So this process is called replication itself. So this is what the central dogma of molecular biology is. Sometimes what happens in some viruses, a reverse thing happens. So we get DNA from RNA, okay? And this is called reverse transcription. And it is an exception to the central dogma of molecular biology. Moving to the packaging of DNA helix. In prokaryotes and eukaryotes, there is a difference in the packaging of DNA helix. So let us see that. So in prokaryotes, we know that the nucleus is not well defined. Okay, so the DNA, which is negatively charged, and few proteins which are positively charged, are just scattered, okay, in the cell. Okay, just like this, and there is no particular distinction. So this part is referred to as nucleoid, and there is not much organization seen here. But in eukaryotes, we see it is pretty much complex. So in eukaryotes, positively charged basic protein constituting of amino acids like lysine and arginine form a 8-molecule complex called histone octamer. Okay, so the spherical structure you see here is nothing but your histone octamer. Okay, around this histone octamer, you can see there is DNA coil. Okay, negatively charged DNA coils around this positively charged histone octamer. So this is negatively charged and it's acidic in nature, whereas this is positively charged and basic in nature. So you see how it is complementary, right? And later, one more protein called the H1 histone, it clips the DNA after coiling like this. So this particular structure, this entire structure is called a nucleosome. So in general, basically a nucleosome has around 200 base pairs wrapped around itself. Okay? And this nucleosome, it appears bead-like balls on the string. So what is this string? Basically, it is nothing but the chromatin. So what happens is that this DNA continues itself, and again one more histone octamer will be present, and it wraps itself around it, and it continues this again. So like this, you will find that it forms a string. Okay, so these are the various nucleosomes present, and the DNA wraps uh, itself around it and continues. So that is why it looks like beads on string. So these are called chromatin. Okay, this is basically what we refer to as chromatin. So chromatin is divided into two types. Okay, we have euchromatin and heterochromatin. So euchromatin is very loosely packed and it is lightly stained, whereas heterochromatin is tightly packed and it stains very darkly. So when the chromatin is tightly packed, replication becomes difficult. That is why it is transcriptionally inactive. That means it does not perform transcription. Whereas euchromatin, it is loosely packed and hence it is transcriptionally active. Furthermore, that we know that basically these chromatin fibers, they condense to form chromosomes during cell division, right? So for this to happen, for the packaging of the chromosome into dense packaging of chromatin into dense chromosome, we need additional set of proteins, an extra set of proteins, and these are called non-histone chromosomal proteins. And they help in the packaging of chromatin during prophase.